Hey there, we're back. 233 endocrine lectures. We're going to be finishing up today doing pancreas and a few other miscellaneous glands. It might be another longer lecture, so feel free to stop in between, take breaks, rewind, look at things as you need to. So let's switch over to where we left off last in the notes. So we are on page 39 in the pancreas. And that's what we're going to spend most of our time talking about today. Pancreas is an important endocrine organ, also has an exocrine part of it as well, and that's what this is saying down here. So where is the pancreas first? It's right behind the stomach and kind of a little inferior to the stomach, and the head of the pancreas is right where the small intestine, the beginning of the small intestine is. And it's going to secrete some digestive enzymes into that area of the small intestine. And we'll talk more about that when we get to digestion in a week or two. That's the exocrine part of the pancreas. And the acinar cells, which are the bulk of the cells that you see in the pancreas, are doing that. However, it also has endocrine gland functions, and this is where those pancreatic islets, where the islets of Langerhans are, and these guys are the ones that you recall from your lab report in the first week. There are three different cell types. There are alpha cells that secrete glucagon, beta cells that secrete insulin, also amylin, and then delta cells which secrete somatostatin, which is also GHIH. So that's kind of always good to remember that that is growth hormone inhibiting hormone. Islets are surrounded by the acinar cells and a whole bunch of capillaries and sinusoids, and actually we can look at one right there. There is one of the islets, and you can see the slightly darker pink stained alpha cells there, and then the lighter stained beta cells, and all around here these kind of bigger pink cells, pinkish purple, are the acinar cells, which are the exocrine function ones. So you've seen that already. We're going to talk about the homeostatic balance and imbalance of blood sugar in a minute, and so we'll go to this figure a little later, so I'm going to bring it up for later. Let's talk about glucagon and insulin, okay? We're going to actually talk about glucagon first. About 20% of the secretion of the pancreas is glucagon, and then about 75% is insulin. Both hormones work to regulate blood sugar levels, and they in effect are antagonists. They work kind of opposite of each other, and you'll see that as we start to write everything down, okay? So glucagon's very potent, and when we have an increase of a secretion of glucagon, okay, so when glucagon is released into the blood, into the interstitial fluid, and then gets picked up by the bloodstream, what it will do is it will cause when glucagon is released, it's released because there are low levels of blood glucose, all right? So when we see low levels of blood glucose, we see an increase in secretion of glucagon. If we have high levels of blood glucose or high levels of somatostatin, which we just said on the previous page was GHIH, right? It'll inhibit it, okay? So glucagon is inhibited by high blood sugar levels and somatostatin, GHIH. Okay, so what is glucagon really? And we'll put a little star here because this is important. Glucagon is a hyperglycemic hormone whose principal physiological activity is to affect an increase in blood glucose levels. So if blood glucose levels are low, glucagon's job is to get blood sugar or get sugar into the bloodstream and increase blood glucose levels. Okay, so how does it do this? Well, glucagon targets certain areas of the body, and it does. It targets these areas, and we need to learn some new terms. All right, so let's look at this first term, glycogenolysis. So we know that lysis means to break down, and we know glycogen, we know, is our storage form of glucose. So we would break down glycogen. So we convert anywhere we have glycogen into glucose. So think about the places where we know glycogen lives: muscles, liver. So we would target those areas and break down the glycogen into glucose, and that would increase blood glucose levels. Now, gluconeogenesis, we talked about this previous lecture. Make new glucose. Okay, so this is where we would produce glucose from non-carbohydrate sources like proteins, amino acids, fatty acids, glycerols, even lactic acid. Remember, we can take lactic acid and convert it back into glucose. So glucagon will help us do all those things. It'll also promote lipolysis. We also, I believe, talked about this one last time. Lipolysis is breaking down triglycerides, fats, into the fatty acids and glycerol, the monomers, and use them as energy sources. So these fatty acids then become an energy source. We also will take amino acids up into the liver, and we will then make glucose from them. So that's another thing that it does. So lots of functions, but basically all of them do the same thing. They increase blood glucose levels. There is a abnormality, a glucagonoma. Whenever there's an OMA at the end of something, it's a cancer or a tumor. So this is a glucagon secreting tumor. And so you can imagine what would happen if we had a glucagon secreting tumor. We would get hyperglycemia. We'd just oversaturate the blood with blood glucose, and we would get a diabetes-like condition, right? So we might develop diabetes as a result of this. One of the symptoms, and it's not really that you have to memorize this, I'm just putting it in for completeness here, is this strange skin rash in the genital region. And this is one way that it could be diagnosed. But not too worried about that. What's more important is how glucagon is going to cause an increase in blood glucose levels. Insulin is going to do the opposite. So insulin's job is a hypoglycemic hormone whose principal activity is to reduce blood glucose levels. And the way it does that is it takes glucose and pushes the glucose into, or transports the glucose into, cells of the body. Okay? So insulin is even more potent and longer lasting than glucagon, and we secrete insulin when we have high blood glucose levels, and it's inhibited by somatostatin, right? So it's also inhibited by somatostatin. Again, that is also known as GHIH, right? So what are some of the major targets? Major targets. Almost exactly opposite of what we talked about with glucagon. And this is one good thing about learning these two hormones, is if you learn one of the hormones, you can kind of, in effect, know what the other one does by just reversing everything that we said. So if you just want to learn about insulin, then you'll know that if there is a question about glucagon, you can just say, oh, well, I just need to reverse everything I know about insulin. So let's look at this. So what does it do? Instead of, so it has a new, basically, as we said, its job is to move glucose into cells for storage as glycogen. And how does it do that? Well, one way is it does this glycogenesis, all right? And this is making glycogen, all right? So instead of breaking glycogen down, we convert glucose into glycogen. We're going to decrease gluconeogenesis, right? We're going to decrease glycogenolysis. That makes sense, because we wouldn't want to break down and make more glucose. We wouldn't want to make new glucose from sources. We want to do the opposite, so we want to break it down. We would increase lipogenesis, all right? And as a result, we would decrease lipolysis. So we would create fat as opposed to breaking down fat. So this, as a real quick side here, is one of the reasons why if you just had a very sugary based diet, you can still put on a lot of body fat. And this is because the sugar is at high levels in your bloodstream. And one of the things that your body does is it tries to put some of that sugar into cells for use. But then those cells are fed completely with sugar and they're still left over. So one of the things that it does is it makes fat. It takes and stores that glucose as fat, and our fat cells then get larger. All right. Also, and one of the things that we do, I guess this is kind of along the same lines, we synthesize those fatty acids in the liver, and they're transported by very low density lipoproteins, which is one of your cholesterol. When you're doing your cholesterols, there's the HDL and the LDL and the VLDL, and they go to the fat cells, and then they're stored as triglycerides, and that's what I was just talking about there. Okay. Now, one thing we will do is if we have a lot of glucose on board, we're going to try to, instead of changing amino acids into glucose, we're going to uptake amino acids and stimulate protein synthesis, especially in muscle. So if you were, say, exercising and you were increasing your glucose in your blood, your body would want to make more muscle, and so then it would do this. So that would be a good thing. Insulin does not affect brain or kidney cells. We want those to stay relatively stable and working. This part here, it says stimulated by the vagus nerve, swollen reflex, and gastrointestinal hormones, and it has all this thing. I'm not even going to talk about that yet. We're going to get to that when we are talking about digestive system. Now, this picture here, this is the pancreas. If you didn't know what the pancreas looked like, pancreas, and it says basically glucagon is released, and it causes high blood sugar, right, to happen, and then we can have a negative feedback on that. We can have insulin cause low blood sugar, and a better picture than this one here is the one that's in your book, which is right here on figure 1617, and that's page 631 in your book. And so normally we'd have a blood sugar level around 90 to 100 milligrams per 100 milliliters. And so if we have a lot of blood glucose, like you eat a really sugary meal, your pancreas will notice this and secrete insulin. And insulin will cause glycogen to form and will store that glucose as glycogen in your liver and your muscle. It'll also target many cells of your body to uptake glucose and use it as fuel. And so it'll try to get rid of that sugar and bring it into the tissue cells. As it does these two things, blood glucose levels will drop and we have a negative feedback loop happening. Now, the exact opposite can happen, right? We said that glucagon is opposite of insulin. So if our blood sugar levels are really low, let's say you are working really hard all day and you forget to eat and 5 or 6 hours have gone by and your blood sugar levels are starting to drop, your pancreas will notice this and release glucagon. Glucagon will cause glycogen to break down, and glycogen will release glucose. And remember, it also will take other sources like amino acids and lactic acid and stuff, and it will make new glucose as well. And those two things will cause the blood glucose to go back up. Okay, so review that if you're not remembering how our negative feedback loops work. That's really great there to kind of figure all that out. So before we go into the abnormalities, the one thing I want to say is there's some new terminology here. Some of these words that we are just looking at. But the nice thing about these words is the words tell you exactly what they're doing. Lipogenesis means creation of fat. Gluconeogenesis means creation of new glucose, right? So glycogenolysis means break down glycogen. So a lot of these things are pretty straightforward. So I think it won't be too hard for you to understand all those things. So what are the abnormalities? Well, one abnormality is just low blood sugar, right? If we get hypoglycemic, right? And this is where a glucagon would get released at some point. But we've all experienced hypoglycemia probably before, where our blood sugar levels are low, and we know what it feels like. We kind of get nervous, dizziness. If it was really bad, you might lose consciousness, but usually you just have that kind of tingly, all over weird body feeling, and your body's kind of telling you, hey, go get some food and try to get your blood sugar level up. So that's more of a symptom and a minor out of homeostasis state. Now, diabetes mellitus is an actual condition, and we're going to talk a little bit about that on the next couple pages here. But let's talk about what it is. So it's a form of hyperglycemia, but it's and hyperglycemia just means an elevation of glucose in the blood, right? Secretion of little no insulin, or that there's not enough insulin receptors, because think, insulin's going to tell target cells to bring in glucose. If they're not working receptors on the cells, then the insulin won't interact with those cells. So either we don't make insulin, or the insulin that we make is not stimulating the cells to do their job. And we'll talk more about that in a minute. One thing to think about in diabetes, where we have this high elevation of glucose, one of the problems is we get this thing called metabolic acidosis. So when glucose can't be used, it gets beta oxidized, so it changes, all right, to there's chemical reaction when glucose can't be used. It gets beta oxidized, and it gets beta oxidized to fatty acids and keto acids. Okay? And so fatty acids go up. So when glucose can't be used, we beta oxidize it to fatty acids and keto acids. All right? Ketones. And I'm going to talk a little more about these keto acids in a second, okay? But when we have any acid increase in our blood, it lowers our blood pH, right? That makes sense. More acids, lower pH, okay? And the ketones are formed when fats or lipids are broken down. This is one of the things. When we break down fats, we get ketones. So if you've heard of keto diets, that's one of the reasons, because they're very fat-focused diets and protein-focused diets and low blood sugar, and a lot of ketones are produced. Now, so because we have a lot of fats in our blood, if we, because of this, all right, we will use a lot of fats for energy. One of the reasons why is if we can't get the blood glucose into the cells, right? So something is not allowing, the insulin isn't working, or we don't have enough insulin, we don't have fuel that's coming into the cells. So we use fat as our fuel more. The problem is then a lot of fats are circulating in the blood, and we get fat deposits on the blood vessel wall. So people with diabetes tend to get plaque formation on their vessels, and can have hardening of the arteries and other atherosclerosis effects. Okay, they also get dehydration. Now, one reason is if you have a large amount of glucose in your blood, and then you're going to try to get rid of that glucose. And one way is you can allow that glucose to go into your urine. But you have to dilute that glucose. All right? You can't just pour bunches of glucose and have this thick, syrupy urine. You have to put a lot of water in with it to dilute it. So as a result of that, we don't reabsorb water. Water stays in the urine, and we get a lot of urine production. Okay? And so we get too much water out of our system, and people with diabetes can get very dehydrated and also get very thirsty, okay, as a result. So they're constantly increasing their water intake. Remember, if we're not using glucose as our main energy source, because it's not going into the cells, we use fats, and we can also use proteins. And so sometimes, especially in later stage diabetes patients, if they're not well regulated, they get muscle atrophy because they break down the protein in their muscles, and their muscles become weak. And then obviously in really uncontrolled diabetes late in the disorder, you can have such low blood sugar and have such problems with the getting fuel to the brain that we can go into a coma, which could lead to death. Now, I did want to mention one thing here about these keto acids, so I'm going to put a little note over here. The keto acids, the ketones, okay, will form a thing called acetoacetic acid. And that becomes acetone. And acetone, you may have heard of acetone before. Sometimes acetone is in certain, like a nail polish remover. And it's volatile, or another way to say that is gaseous, right? It goes into the air. And people who really have uncontrolled diabetes will produce this acetoacetic acid, and they will have some of it convert into acetone. And because it's gaseous, it will come out in their breath. And so you can actually smell on the breath, and it has this kind of fruity, nail polishy odor. And early diagnosis of diabetes, this is one of the ways. They actually did a couple things. One is they would taste the urine to see if the urine was very sugary, because a lot of the blood glucose couldn't go into the body cells, and so it went out in the urine. Another way is they could smell this kind of fruity nail polish odor on the breath. And so those were some of the early diagnoses that they would use to determine whether someone had diabetes mellitus. Now, there's 2 pages here. These pages are very detailed, and I do not want you to focus too much on everything here, but these are great to read and understand. I'm going to highlight just a couple things on these, but you can read through these all you want. But there are 2 different types. There's insulin-dependent diabetes type 1 and non-insulin insulin-dependent diabetes type 2. Okay? And I'm going to talk about those more in a minute on the next page. I do want, though, to talk a little bit more about some of that atherosclerosis that we were just talking about. One thing that can happen is this thing called glycosylation. And in glycosylation, the combination of glucose and amino acids, they get deposited in the capillary walls, and this thickens the basement membrane of the capillary walls, which makes permeability worse. So if permeability is worse, movement in and out of capillaries is impaired, and nutrient delivery and waste removal is impaired. So this is another one of these damaging effects of too much glucose circulating in the blood and not going into the cells. It can cause damage to the tissues that are in the capillary walls, and that's not good. So that was one thing I wanted to note there. And the basement membrane thickens, and it decreases the permeability to nutrients and oxygen. Okay, so that's really what is happening there. Okay, now this one I want to spend a little more time on. This guy is a good overview, and again, it's a little more wordy than it needs to be, but it's a good overview of the 2 types of diabetes. Okay, so diabetes mellitus is hyperglycemia, but it's due to either a deficiency or absence of insulin, which is called insulin deficiency, or the reduction of the effectiveness of insulin. So in the first one, you don't make insulin at all, or you don't make insulin that works. In the second one, you make insulin that should work, but your body has become not sensitive to it anymore and is resistant to the insulin. Okay? The first one is type 1 diabetes, insulin-dependent diabetes mellitus, which means you need insulin, and if you got insulin into your body, things would work normally. The second one, type 2, is non-insulin dependent, which is that your insulin is actually being produced and could work, but your body has stopped reacting to it. Okay? So type 1 diabetes is seen more in young individuals and is occurring more in non-obese adults. Type 2 tends to be having to do with obesity. All right? And in the absence of insulin, target cells will fail to take up the nutrients from the blood and continue to add glucose, amino acids, and fatty acids to the blood from their storage sites. Remember, we can't use glucose as fuel because the target cells don't take them up. So we get we use amino acids and fatty acids instead as fuel. Okay. What we think is happening in this case with type 1 diabetes is that there is some sort of insult to the beta cells, and you usually have to be genetically predisposed to even have this happen. So first you have a genetic predisposition. Then there's something that happens to the beta cells, like it could be a virus or it could be a toxic chemical. And then once that happens, then we get an immune deficiency that leads to destruction of the beta cells. And once those beta cells are destroyed, then we don't produce the insulin anymore. All right, so that is what is happening there. So that's often why it's in younger individuals also, because sometimes early in life they have one of these environmental effects and they stop producing insulin, and it gets noticed, and then they become insulin dependent. Okay, so that's the main thing I want to talk about, that one. So what's the treatment for that? Well, we obviously give those patients insulin. They monitor their blood glucose levels, and if their blood glucose levels get too high, they inject insulin into their body, and that will then allow the cells to uptake the insulin. And people are doing very well and can be very well controlled using all of the different things that we have, injectable insulin pumps, things like that. Now, type 2 diabetes is much more, is a much different disorder and is becoming more and more seen in society. It used to be mostly in adults, but we're seeing more and more individuals, kids get it, and this is usually due to obesity, unfortunately. And what's happening here is you are making insulin, okay? But for some reason, the body stops responding to insulin, okay? And there's two major mechanisms as to why the body stops responding to the insulin. So one is chronic overfeeding, and this would be along with obesity. So what happens is if you are putting lots of blood sugar in, a lot of sugar in, which increases your blood sugar, we're going to continually stimulate the beta cells. Well, over time, they're going to get stimulated so much that the beta cells, and there's so much sugar available, the target cells are going to downregulate and decrease their number of insulin receptors because they're being overstimulated with sugar. So your body cells are basically saying, hey, you're putting too much sugar into our blood. We don't want that much. And so they actually downregulate the number of insulin receptors, and that then results in a decrease in insulin sensitivity in many of the tissues of your body. The bad part is then your blood sugar level stays high. But it's your way of your body cells saying, hey, enough is enough. Too much sugar is coming in. Now, also, it could be that there's some defect in the receptor interaction with the insulin that reduces the ability to clear nutrients from circulation. So imagine like if our storage deposits are too full, we can't do a good job of kind of clearing the rest of the sugar out because we've filled up all of our places we normally store glucose, and as a result we just keep blood sugar levels really, really high, and we just can't take any more in. So in both of those cases, one way we can stop this is to reduce overfeeding, right? So we try to get a better diet, a healthier diet. We decrease the amount of food intake, especially sugars, and that would help restore insulin receptor sites to normal levels, makes insulin more a regular amount of sensitivity, and things can be reversed. And so that's great because this type 2 diabetes can be reversed through diet, exercise, and changes, and hopefully if you do it early enough before there's damage to your body. Where unfortunately in type 1 diabetes, because it's a genetic predisposition disorder and the pancreatic beta cells have been destroyed, we can't reverse that. Maybe in the future we'll be able to do something where we inject new beta cells into the person or something. But for now, we can't do that. So that's an important thing to note, the difference between the two. And so review those pages, and hopefully that will help you understand a little bit about them. Now, we're almost done. We only have a couple more things to talk about. We've gone through almost every gland in the body. We have the gonads and the pineal gland and the thymus. Those are our last three. And these we're going to do really quickly. We're going to spend more time with the gonads when we get to reproductive system. So you're going to see me hardly underline anything here. So what do the gonads do? They produce hormones, and they also obviously control reproductive activities. So the ovaries, the female gonads, they produce progesterone and estrogen. And that the job of then the ovaries is to regulate the development and maintenance of female secondary sex characteristics. And the big thing that ovaries do is they do allow production of and maturation and production of oocytes, eggs, and allow reproduction to occur. That's it. That's all we want to talk about now. Testes, male gonads, right? And in testes, they produce testosterone, the primary male sex hormone. And guess what? It does a very similar thing. It regulates the development of male secondary sex characteristics, and it produces sperm. Okay, and maintains reproductive organs in males. All right, pineal gland. Now the pineal gland, you might remember way back from 231, we had the epithalamus, which was also called the pineal gland, and it was right behind the thalamus there. And you might recall that, and it's kind of a pine cone shaped, which is where the pineal comes from, and it's at the roof of the third ventricle, and right there next to the thalamus. And what it does, or to kind of hint at what it does, is secretions peak between the ages of 1 to 5, and most of the secretions have decreased a lot by the end of puberty. And what it secretes is melatonin. And melatonin, we know, and we think about, it helps with our circadian rhythms, which is a way to say our sleep wake cycle, right? And this is kind of opposite of it's secreted kind of opposite of cortisol, where melatonin levels go up in the afternoon and evening and make us sleepy, and then in the morning they start to go down and cortisol goes up, and the combination of those two starts to wake us up. All right. And one thing that's interesting, and it's a little unclear, is melatonin during these early ages appears to inhibit reproductive activities by inhibiting gonadotropic hormone release, and it inhibits early sexual maturation. It's a little, and as I said, I'm going to say this is a little unclear. We're still doing research on this, but that kind of suggests why we see a lot of melatonin early on, and then it drops off by puberty, because it seems to kind of try to forestall early sexual maturation as well. Okay. Our last one, thymus. Now the thymus gland, you can see where it's located. It's below the thyroid gland there. It's deep to the sternum, and it's very large in infants and children, and it goes away as we get older. And you'll notice in a minute why it goes away when we get older, because so in adults, it's mostly fat and fibrous connective tissue. It's reduced in size. And the reason why is it is involved in immunocompetency. It develops and activates T lymphocytes and is essential for normal development of an immune system in children. One thing that I've said before about it is that it is kind of a safe place for the T lymphocytes to practice being T lymphocytes and try to get them to work the way they're supposed to. There are hormones that's secreted here, thymopoietin and thymosin. These guys, oops, sorry, my pen It's not working all of a sudden. There we go. Are the hormones, and these are what stimulate the T lymphocytes for for activity. Interestingly, some of these hormones are being used in patients that need to increase their T lymphocyte activity, like cancer patients, for example. So there's companies out there that are making thymosin and trying to inject it in adults because we don't have much of it, but it might help us with and make our immune system better. Okay, I'm going to pause for a second, but we're not going to go away here. I'm going to bring something else up on the screen, so I'm going to go away for a moment, come right back. Okay, I'm back, and I am not on our lecture notes anymore. I am on a PowerPoint slide. I'm going to put this PowerPoint slide on the Moodle if it's not out there already, and it says metabolic syndrome. And metabolic syndrome is kind of a good way to bridge between the endocrine system and the digestive system, which we're about to get into. You may have heard of metabolic syndrome before. It's a syndrome, as this slide is saying, characterized by a group of risk factors that occur together. And when you have certain levels or numbers for these risk factors, they increase the risk for both coronary artery disease, stroke, and what we were just talking about, type 2 diabetes a few minutes ago in the lecture. So what are some of the risk factors? Well, you can see abdominal obesity, basically the size, diameter of your measurement of your abdomen is a risk factor. We'll talk about that in a minute. High triglycerides and low HDL. HDL is the good cholesterol level, so that's another one we look for. Hypertension, high blood pressure, insulin resistance, which is what we are talking about, is type 2 diabetes, where our body starts to not react to insulin in the right way, and then also a pro-inflammatory state, meaning that our body is starting to get inflammation due to some of these problems, and that inflammation continues. So the next slide gives the guidelines from the American Health Association, I believe. So in males, if your waist circumference is more than 40 inches or 35 inches for females, you're at risk. If your triglycerides are above 150 and your HDL is below 40 for males or 50 for females. Hypertension, you can just be slightly hypertensive, about 135 over 85 or higher would be a risk factor. And if your fasting glucose is above 100 milligrams per deciliter, can also, or per 100 milliliters, as we were using earlier, can be a risk factor. So what is the incidence and implications? And this is a few years ago. This is probably higher now. If someone wants to go out and research what the metabolic syndrome incidence is these days, you can. I probably did this about 3 or 4 years ago. About 1 in 4 Americans and about 40% over the age of 40 have metabolic syndrome. So we're talking about 50 million people. This increases their risk for coronary heart disease and other inclusive diseases. It increases the risk of type 2 diabetes. But there are things we can do, just like with type 2 diabetes, that can help prevent or reverse the disorder. So first is healthy weight control. If you can get your weight down, then that'll be much less stressor on your body. If you can manage your blood pressure, your lipid, and your glucose levels as well as possible, excellent. If you're a smoker, stop smoking. The better your nutrition, the better, especially increasing fiber and less sugar and bad fats, but better things that have lots of fiber and lots of fruits and vegetables, for example. And then moderate to vigorous exercise multiple times a week. The more, the better. The AHA is American Heart Association. I was going to say also the American Heart Association recommends, I think their recommendation is currently, trying to remember the number now, somewhere between 75 and 90 minutes of moderate to intense activity a week, and something like 150 minutes of low intensity, like walking, a week. So if you walked every day, 30 minutes a day, 5 days a week, you'd probably meet that criteria. If you went for a 3-mile run 3 days a week, you would probably approximately meet that criteria. So we're not talking about huge exercises, but something that any amount, and the more the better, will help stem off metabolic syndrome. Now, one other thing, I believe on the Moodle site right now, I have a couple of videos about metabolic syndrome. They're a little bit older, but they're useful. So watch those. There might be a question or two about metabolic syndrome on the exam. And even if not, because a lot of it has to do with the risk for type 2 diabetes, learning about it kind of helps you understand a little bit about insulin resistance and type 2 diabetes as well. All right, so we are done with endocrine. Endocrine is a big chapter. We spend a lot of time on it, multiple weeks. The other thing I want to note is in your notes, and let's see if I can switch over to them. I'm not sure. I can get over there here real quickly. In your notes, you're going to notice that we have a lot of study aids in the back. So we have all the hormones we've talked about listed, where they are, what they're stimulated by, what they're inhibited, what their targets and actions are, what diseases are resulting. Those are really good and useful. You can see lots and lots of them. These pictures are probably less useful. They really show kind of just how complex everything is, but I don't think they would be helpful for you to memorize or study, but you can look at them. These little guys are nice because they're like little flashcards almost, or little summary cards for each one. And one thing that we sometimes suggest is, as you're learning these hormones, what you do is you want to be able to kind of fill out this kind of information, okay? And so if you look, and I'm going to scroll down to the bottom here, at the very end of all of these, we have some copies of these. So this little section right here, from here to here, is one, and then on the page we have a second one. So you could print this page out and print out a couple. And so what you could do is you can go through and you can type in a hormone name, okay? So you can say, something like oxytocin, okay? And then it says location or released from, and so then you might say oxytocin is made in the hypothalamus. You can give the specific nucleus and everything if you want. And it travels down the HH tract and is stored in the posterior pituitary. So you could write all that in right there, something like that. And that could be the mechanism of release, which is neural stimulation. So we could say here, neural stimulation causes the release of oxytocin, and then you could talk about what the neural stimulation is. So you could say, pressure on the cervix and the uterus. You could say stimulation of the nipple. So those would be the two neural stimulations. Then we'd say negative feedback system, and you'd say no, cross it out. You'd say oxytocin is a positive feedback system, and you could explain what that's about. Then you would say, where does it target? And we would say, it targets the uterus, and it targets the breast tissue. And then you could say what the actions are of that. And then if we mention any diseases or conditions, you could put those in as well. So this is a good way to practice. You write in a hormone up here, and then you write as many things about how it's released, how does it go through the portal system or through the tract, what stimulates it, and goes all the way down, okay? That's a really good way to do it. If it has a releasing hormone or an inhibiting hormone, we want to talk about that in the negative feedback and in the mechanism of release. So this is a good way to practice. I encourage you to do that. There may be some questions on the exam where we'll take a hormone and we'll ask certain questions about how it's released and what happens and all these things. And so we'll probably do that for a couple different hormones. And so it's nice if you can fill these out to then be able to recall those and answer them on the exam. Okay, so thanks for another long lecture, listening to another long lecture, but we are now done with endocrine, and next week we'll be starting with digestive system. So as always, if you have any questions, send an email or ask your instructor, and I hope you have a good week. Bye.